Well, it truly has been a long and winding road. So moving on to the next segment of this journey, our next speaker is a professor of pediatrics at Johns Hopkins School of Medicine, where he did his medical school work and stayed on as a resident before moving to Cincinnati, where he worked with Jeff Whitsett and others, learning both to be a neonatologist and how to do basic science. And then he moved to Washington University in St. Louis for 4 years, where he worked with the likes of Sesh Cole and Harvey Colton. And then he moved back home to Johns Hopkins, where he spent the rest of his career. As David commented, he had a seminal paper describing the clinical and molecular underpinnings of surfactant B deficiency in 1993, and has spent the rest of his career really cataloging both the molecular and clinical characteristics of disorders of surfactant, which he'll be telling us more about at this point. One of the things that's been really remarkable is that these tests we now sort of take for granted. We write an order, fill in a little information sheet, and this testing then goes off to one of a variety of commercial laboratories that make it available. But in the early days, we had to send this testing to Dr. Nogee's lab. And the great thing about that was that we not only got a molecular analysis that told us whether there was a mutation or not, and if there was, what it was, but we got to have a phone conversation with Dr. Nogee, who very generously shared not just his molecular expertise, but also his clinical expertise in helping us figure out how to manage these babies. And even if the baby turned out not to have a molecular genetic disorder, Larry was an enormous help to many of us, and I can personally testify to having been the recipient of his generosity and his wisdom many times over those early years as we learned how to take care of these diseases. So it's a great pleasure at this time to welcome Dr. Larry Nogee to the podium. Thank you, Larry.
Well, thank you for that very kind introduction. It's a great honor to be here this morning. I'm also very humbled by Dr. Kornfeld's introduction and listing my paper up with so many others that I admired. It's been 25 years plus since we first learned about SPB deficiency, and what I want to do is give you a little bit of an update on sort of where we are with these disorders. The background of the slide here is not what I see from my backyard in Baltimore, but there is a link here. This is a beach from the Cape Cod National Seashore. Dr. Kornfeld asked us to talk about our journey to discovery last night. I was on my first vacation on Cape Cod in 1963 in August, and the Kennedy baby was actually born on Otis Air Force Base. So this was very much in the news and in my conscience and made a big impression on my 8-year-old brain. The other thing, are there any trainees in the audience? So if you go back to that New York Times article on the 50th anniversary of the Kennedy baby, one of the striking things in there is when the call came in to Boston Children's, there were no attendings in-house. It was a senior resident who had to take the call. So that's something to remind your trainees when they're feeling left alone. So Dr. Sunshine did a wonderful update on RDS, and lack of pulmonary surfactant due to immaturity is the principal cause of RDS in premature infants. This is the standard pie diagram of what surfactant is made of. It's not a simple thing, it's a complex mixture. That's predominantly lipid, and the predominant lipid is disaturated phosphatidylcholine. And it's DSPC, which is the major ingredient that helps lower surface tension. It's thought to do so by its ability to form a monolayer of the air-liquid interface with the positively charged moieties pointing towards the liquid surface and the palmitic acid tails pointing to the atmosphere and be able to be very tightly compressed. However, the enzymes for making DSPC are not lung-specific, and how DSPC becomes concentrated in surfactant wasn't immediately obvious. More importantly, DSPC by itself does not absorb very quickly to an air-liquid interface. This material isn't dumped on the way it is when we treat babies with RDS, but it needs to transition through a thin layer of liquid from the alveolar cell and spread at the air-liquid interface. And that's where these proteins come in. The 10% of surfactant that is protein by weight contains mainly serum proteins, but also contains some relatively lung-specific proteins, of which two very small hydrophobic proteins, surfactant proteins B and C, play absolutely critical roles in enhancing the surface tension lowering properties of surfactant lipids. Now, we've learned about SPB deficiency about 25 years ago by hypothesizing that perhaps full-term babies who presented with an RDS phenotype would be a group that would be enriched for genetic mechanisms. And this turned out to be the case. While that was a hypothesis, it was also fortuitous in terms of the baby coming to us in order to be able to test this hypothesis. As it turns out, while this may be the first discovered disorder, this is also the rarest. We have data now from population frequency of mutations that this is less than 1 in a million live births in the U.S. So this is an extraordinarily rare disease. It is, however, an unfortunate one. These children present with an RDS-like picture, clinically and radiographically, but unlike preterm babies who either, if you can support for a week or treat with surfactant to get better, these children have persistent disease and will die from their disease. It's an autosomal recessive disorder. The mechanism is biallelic loss of function, mutations in the gene encoding SPB. The pathology is interesting in that there's this accumulation of this granular material filling up the distal air spaces, and this is what was called alveolar proteinosis. It's perhaps unfortunate that we use this term because the pathophysiology of alveolar proteinosis in older children and adults is very different. But it was a very striking feature and pointed us towards a disruption of surfactant metabolism. If you do electron microscopy, you'll see these are normal lamellar bodies here. These are the storage organelles for surfactant in a type 2 cell. And in SPB deficient children, they do not have normal lamellar bodies. Instead, they have these very disorganized organelles that are filled with multiple vesicles. So SPB is needed intracellularly as well as extracellularly in order to package surfactant. And this is a terrible disease. Almost all of the children die within the first 3 months of life. There are some rare survivors who can survive longer because they have mutations that allow for a little bit of SPB to be made. Now, this pathology may have sort of been misguided us a little bit in terms of terminology, was helpful in terms of sort of thinking about what other children should we look at for other potential mutations. So obviously we wanted to learn about SPC, but as it turns out, SPC deficiency, and I've used the term dysfunction instead, doesn't result usually in neonatal disease. This is also a very rare disease. We really don't know its incidence and prevalence. Clinically, while they may present with neonatal RDS, this is an exception. This is less than 10%. Most present early in infancy with what's called childhood interstitial lung disease. Here's a CT scan here showing diffuse ground glass infiltrates, or they may present even as adults with pulmonary fibrosis. The pathology has a number of different terms: chronic pneumonitis of infancy, desquin interstitial pneumonia, nonspecific interstitial pneumonia. But you see that there are these accumulations of macrophages and sometimes proteinaceous material in the air spaces, along with very prominent type 2 cell hyperplasia and a very thick interstitium. Unlike SPB deficiency, this is an autosomal dominant disorder. You need a mutation on only one allele, and these are coding mutations in the SPC gene that affect the structure of the proprotein. The disease can also occur sporadically due to de novo mutations. There is a common mutation that's accounted for about a third of the cases. It's a substitution of threonine for isoleucine encoding 73, but there's no real genotype-phenotype correlation here. We have patients with the same mutation within the same family who have very different presentations. The mechanism is opposed to a loss of function, a gain of toxic function, and it is somewhat dependent upon the location within the protein itself, and it's a complicated mechanism that has to do with protein folding, mistrafficking, but basically making a protein within the cell that is toxic to the cell. Now, one of the new things we've learned, and I think one of the themes for this morning, is every time we think we understand something about these diseases, we're learning something new, is that while we've thought that this is exclusively a dominant disorder, it turns out that about 5% of the patients actually have mutations on both alleles. The natural history is highly variable in its onset and severity, and the other thing we're realizing is that even infants who are critically ill in either the newborn period or early infancy and on ventilators for prolonged times can show significant spontaneous improvement, maybe due to the treatments that we throw at them, but that the course of the disease does not have the trajectory of SPB deficiency. So this is an example of two families who share the same mutation in SPC, a substitution of leucine for proline in codon 14. But the pattern here is one of an autosomal recessive. The parents are well, but the children who get both mutations are the ones who develop disease. Interestingly, these two families are geographically quite remote, but still share a similar ethnicity, so this is probably a founder effect mutation. And as we've gone through, we found actually a number of other families who have, again, biallelic mutations, sometimes with consanguinity and sharing the same mutation, but some who are compound heterozygotes as well. So this is not exclusively a monoallelic disease. And interestingly, some of the newer mouse models developed by Mike Beers at University of Pennsylvania, you really need to have both alleles affected in order to fully manifest disease. So it may be that the critical link here is that there needs to be some second hit, either environmental or genetic. These are outcomes of children with SPC mutations that we have gathered over the years. These data are from 10 years ago, when we had about 6 or 7 years experience. You can see that some infants and children died, some had lung transplants, but we had a fair number of survivors, including adults, without any symptoms. We update this sort of same database 10 years later, and what we're seeing is that there's, while still a little bit mortality, we're seeing a lot more sort of survivors. And this has been reported by Robin Dieterding as well, that even critically ill neonates and infants can improve with time. And this has made the decision of how to manage these infants and when and if to refer them to lung transplant very difficult, because there is such capacity for improvement. ABCA3 deficiency is perhaps the most common of these disorders. The protein stands for ATP binding cassette member A3. This is a member of a large family of membrane transporters that move substances across biological membranes. The one you're most familiar with as pediatricians is CFTR, also known as ABCC7. So this is a cousin of CFTR. But ABCA3 instead sits on the surface of lamellar bodies. And its role seems to be to import those lipids, particularly DSPC and phosphatidylglycerol, that are critical for surfactant function into the lamellar bodies. This is shown both by mouse models in which you can look at the lamellar body composition as well as in lung lavages from human infants who've undergone transplantation. We don't really know the incidence. I'll show you some data in a few minutes. Maybe as high as 1 in 4,000, that's probably an overestimate, and 1 in 20 is probably more realistic. These children can present identically to children with SPB deficiency, or they can present with childhood interstitial lung disease, or even as adults. This is again a recessive disorder. You need to have biallelic loss of functions on both alleles. Complete loss of function mutations predict a poor outcome, but monoallelic mutations, heterozygous or single mutations, may also increase the risk of RDS, particularly in late preterm infants, and may be susceptibility alleles for interstitial lung disease. The pathology is similar to that of both SPB deficiency and SPC, with proteinaceous material, accumulation of macrophage in air spaces, type 2 cell hyperplasia. The EM is different. Instead of sort of these nice, well-formed lamellar bodies, what you see these small, dense bodies with the core pushed off to the side, giving them this so-called fried egg appearance. And the natural history here is that about 60% have a lethal disorder in the newborn period, but we're recognizing increasingly that some children and adults can survive much longer. This is a much larger gene than either SPB or SBC. It's got 30 coding exons on the 16th chromosome. We know of over 400 mutations. It's a little hard to keep track. And there are all kinds of different mutations that are scattered across the gene, including some repeat offenders, but no real predominant mutation. One of the other important things is we've recognized that there are multiple disease alleles with more than one mutation on the same allele. So it's very important if one has a patient with this disorder and you get back a report that shows that there are two mutations, that you make sure that both mutations are on opposite alleles rather than on the same allele before concluding the child's affected. We also know that there's at least one deep intronic mutation, and there are probably others that are regulatory, so that are accounting for some kids that we're missing. And we're also, in the past couple of years, we're now recognizing that there are large deletions within this gene, which can be missed if you are just doing routine sequencing of the gene. The genotype does help predict the outcome. This is a retrospective study we did a couple of years ago in collaboration with Alicia Casey at Boston Children's and Jen Wambach at WashU. And what we showed here is that any child who had what we called a null allele, so that was either a nonsense or frameshift mutation, which would be predicted to completely preclude any ABCA3 expression or function, all of these children either died or were transplanted within the first year of life. And the children who survived were children who had other kinds of mutations that presumably allowed for some functional ABCA3 to be made. So what is the incidence of this disease? Well, Jen Wambach at WashU did a case control study of infants greater than 34 weeks with and without RDS. And what she found is that 3.7% had monoallelic ABCA3 variants. If you looked at the RDS group, it was enriched significantly. It was 15%, and hence this increased risk for RDS for late preterm infants, which has since been replicated by Jeff Murray's group. But if you do the calculations, this comes out to a disease incidence of 1 in 3,000. Well, are we missing lots of kids? I don't think so. I think awareness is now pretty good for this disease. Or are some of these variants not truly pathogenic? Well, if you look at some of these variants, and they're listed here with the single letter abbreviations, and the E292V is clearly a pathogenic mutation. We know of over 30 subjects with lung disease and a different pathogenic mutation in trans. These two are a little less clear. These are the number of alleles in a very large database. These are the number of homozygotes. Both of these do seem to increase some risk. So Jen has taken these and looked in cell culture at what happens. And this is looking at ATPase activity, and what you see is that this R288K variant and E292V, as well as an unrelated variant, all do reduce the activity of this protein. So if you look at a model for what's happening, here's a wild type on both alleles, here's someone who's a carrier, and if we assume that there's a critical level of this protein needed for normal lung function, then what you see is if you have this mutation, which is relatively frequent, and a null allele, then you're going to be below this threshold and have disease. But if you're homozygous for this mutation or some of these other relatively milder ones, you're going to be either above that threshold or just at it. So that we think that these then maybe confer some susceptibility if there's the right either genetic or environmental context to reduce ABCA3 function further. A fourth gene which we've recognized in the past several years can cause an overlapping phenotype is NKX2.1. This encodes the transcription factor, thyroid transcription factor 1. In the lung, this turns on lots of genes, including ABCA3, SBA, SBB, C, and several thousand others. There are lots of different variants that have been identified, including very large deletions that take out other genes in the region, and the mechanism seems to be haploinsufficiency, or losing the function from one allele. The phenotype is not just confined to the lungs. This protein is, as the name implies, critical for thyroid genesis, as well as important for central nervous system development. So it's called brain thyroid lung syndrome. In the brain, it's primarily movement disorders, but in young infants, it can be hypotonia, which can be difficult to discern if the child is sick from lung disease. And it's a Venn diagram of sort of the overlap. About almost half the patients have manifestations in all three organ systems, very rare cause of isolated congenital hypothyroidism, and 7 to 10% with isolated lung disease, with a huge caveat that there's a lot of ascertainment bias in how these patients are identified and studied. We think that the pulmonary phenotype may be mediated by, in part, some of the downstream genes that are most affected. So what's shown here is immunohistochemistry control on the left, a baby who died with an RDS phenotype in the newborn period, and a baby who presented after a severe influenza infection and then developed respiratory failure. All these two children both had mutations in their gene for NKX2.1, but what you'll see here is this child with the RDS phenotype has absent staining for ABCA3. This child has absent staining for surfactant protein D, which is one of the pulmonary collectins and is important in innate immunity, and particularly in viral infections and influenza. So we think that the pulmonary phenotype is mediated largely through the downstream genes that are dysregulated. The gene does matter in terms of outcome. These are survival curves from some of the data that we've collected over the years for the different disorders. You can see that SPC has some mortality, but by and large, about 80% of these individuals are still alive into adulthood. SPB is generally rapidly fatal, with some rare exceptions. And then ABCA3 has significant early mortality, but then a large number of these individuals can survive. How do we diagnose these disorders? Well, what about looking at tracheal or bronchoalveolar lavage for surfactant components, particularly SPB? Well, we've done this over the years, and the specificity and sensitivity are low. There's considerable overlap. Children with ABCA3 deficiency can also have SPB levels that are undetectable, as can children with SBC. Moreover, these tests aren't commercially available and are unlikely to ever be commercially available. What about lung biopsy? Well, it's certainly good for excluding other disorders, and you can get a suggestion of the diagnosis. There are some characteristic findings that can point you to the diagnosis, but they're not gene-specific. So here's an example of child with SPB, NKX2.1, SBC, and ABCA3, and you can see that there's considerable overlap in the lung histopathology findings. EM can be helpful, as I've pointed out before. Here's a SPB deficient child with these vesiculated lamellar bodies in the fried egg and ABCA3. Lung biopsy is also invasive and has substantial morbidity. So genetic testing has become the preferred method. It's got the pros that it's non-invasive, you can get a specific diagnosis, it's commercially available, and the specific gene involved is important for prognosis and recurrence risk. The negatives are the costs, it's expensive, and the turnaround times, which from commercial labs is in the range of weeks, and that may be too slow for a critically ill infant. It's also not 100% sensitive. We have mutations in non-coding regions and unknown genes. And then the real fly in the ointment are the specificity, the variants of unknown significance. So these are some now clinically available panels that test for these disorders as well as others. This is only a partial listing. You can go to the NCBA website, put in gene testing registry, and come up with 40 different laboratories. You can see there's a wide variety in the number of genes, whether they include copy number variants, deletions or duplications, the cost, and the turnaround time. And I've put on the bottom here whole exome sequencing. So in terms of comparing genetic testing versus lung biopsy, this is a thought experiment. So if you were to have 100 consecutive patients that you suspected one of these disorders, and to biopsy them first, and then only do the genetic testing on the ones with suggestive pathology, this is the cost that you would sort of expect. If you were to do the genetic testing first for a 5-gene panel for the most likely genes, you see that when you have about a 7.5% incidence here who have a genetic cause, it becomes more cost-effective to do the genetic testing first. Interestingly, that was the positive hit rate in a UK study. If we change a little bit, we now do a more comprehensive panel. It will cost more, but it will also hopefully pick up more. And then the cost is also decreasing. This is a newer panel, which has about a third of the cost of this one. And then I put here for comparison, if one did rapid whole exome sequencing, that this is clearly more expensive. On the other hand, if you look at some of the studies that have come out from Steve Kingsmore's group in critically ill infants, 30 to 40% is not an unrealistic yield. So whole exome sequencing is sort of where the field is moving. How can we enhance the yield of genetic testing? So we've had a little fun. We've done a retrospective analysis. I've gone back to my database and said, okay, let's pick out some babies with SPB or ABCA3 deficiency. I've got a limited number in which I've got follow-up. I know the babies got better, had uncomplicated course, and being a neonatologist, made a little bit of equivalent of an APGAR score here based on some clinically identifiable risk factors, gestational age, route of delivery. And this is the histogram that you see of the scores. And it's reasonable. By the time you get to a score of 6 or so, you're not going to get all the kids, but you're starting to get a reasonable specificity. So this may help guide us. And I think what we already know about clinically, it's the term baby without any risk factors, delivered vaginally, who's got persistent disease, who's likely to be a candidate. So what are some of the cons of genetic testing? Well, this is a baby from our unit who was born at 410 grams, 26 weeks, had severe RDS, BPD and pulmonary hypertension, ended up trached and ventilated for 20 months, ended up getting transferred to the PICU. This is the x-ray here at about a year and a half of age. It's pretty ugly looking. And they sent genetic testing, not after asking me. And what they came up were 3 variants of unknown significance in ABCA3, as well as a couple in some genes for Hermansky-Pudlak. One of these is predicted to be pathogenic. We weren't able to get DNA from the parents, and we really don't know whether this child was affected or not. I suspect not, but this is the conundrum. And my word of caution is to think very carefully before testing very low birth weight babies, because the prevalence of variants in ABCA3 is such that you're much more likely to get an ambiguous result in an extremely preterm baby than you are a definitive result. So what else is sort of new? Well, when we think we've sort of figured things out, something else comes along. So this is a family that I interacted with some 10 years ago. Unfortunately, it was a sad sort of story. This child died of chronic pneumonitis of infancy. The father developed pulmonary fibrosis and lung cancer and died shortly after this child was born. We did studies, and they weren't really all that helpful. It turns out the mother contributed an ABCA3 variant. The father had an SPB variant, an insertion of a complete codon, and both children had this bigenic inheritance. So we wondered, and we did the immunohistochemistry. This didn't look like SPB deficiency. We also sequenced the SPA genes because SPA mutations have been associated with lung cancer and didn't find anything. And we wondered, is this a sort of oligogenic disease inheritance, a hit in ABCA3 and SPB, and that's what caused this. Well, time goes on, and this variant's not in the public databases, but we now have 3 other children with the same variant. Interestingly, one is an infant who presented following a viral infection, had typical pathology, but no EM. This family then got lost to follow-up. And the reason they got lost to follow-up is because they were dealing with the father's lung cancer diagnosis. And then there was a late preterm infant, again with severe RDS, persistent disease. Mother had chronic lung disease, and multiple other relatives with pulmonary fibrosis and lung cancer. And then last, a 2-month-old girl who was previously well, respiratory failure following a viral infection. And in this case, they did whole exome sequencing. A trio looked at both parents, found the same variant absent in both parents, so de novo suggested this is the cause of disease. So when we think we've got things figured out that SPB is a recessive disorder, here we have what's likely a gain of toxic function mechanism. Lastly, let's talk for a few minutes about therapy. This is for SPC, and this is from a paper from the German group showing some of the things that we throw at them: steroids, hydroxychloroquine, with some of the children having a good to moderate response, but none of this having been really subjected to good randomized controlled trials. Lung transplantation's an option. This is from the WashU group. They published their long-term results a couple of years ago comparing two epics, and it's feasible, even in infants, although it's limited donor supply. Obviously, this has lots of consequences, costs, immunosuppression, and then long-term complications. And we try and tell the families that this is not a cure, but replacing one disease with another. But what's on the horizon? So this is work from Darrell Cotton's group in Boston, and they take tissue, blood cells from a newborn with SPB deficiency. And here you can see the pathology in the EM. And they were able to derive induced pluripotent stem cells. And here's the genotype here. They then corrected it in vitro with CRISPR-Cas, and they were able to differentiate to drive these cells forward. And here's the abnormal cells with the same EMs similar to the human, and here they are corrected. So showing that at least in the test tube, we can correct this and drive things forward. I think also very importantly, this gives us a tool with these alveolar spheres that he's developed that recapitulate the phenotype to have some in vitro tools to move forward. And I think where there's some real hope, this is from the German group. So they are trying to rescue ABCA3 mutations similar to what's been done with CFTR. And here they're using a simple assay looking at processing. This is wild type. The closer you are to one is normal. And here are various mutations. These compounds are proprietary. But what you can see is that for some of the mutations, they can get the expression up pretty good. So the hope is that we'll be able to take some of these same medicines that have been used for CF, which is, again, an ABCA protein, and to be used to treat these for some of the children with ABCA3 deficiency. So then this is just a summary slide going through these 4 disorders with the different sort of phenotypes here. Some of the newer things in red. We used to think that SPB was completely recessive, but we now know that there's at least one dominant allele. The monoallelic risk for RDS for ABCA3. The pathology here is overlapping. Gail will talk later, but I think one of the things we think is probably the specific pathology may be more age-dependent than it is gene-dependent. And then the treatment approaches here, lung transplantation being an offer, an option, currently mainly medical and supportive, and that knowing the genetic cause is important because it informs both prognosis and recurrence risk. And I'll stop there. I need to acknowledge all the many people who've helped me over the years with this. It does take a village. It's not just me. It's given me the privilege to help lots of families and also to interact with lots of people over the years, such as Dr. Bennett's, and I've learned from those conversations as well. Thank you.
Very good. 1 or 2 quick questions or comments for Dr. Nogee? If not, well, David?
Could I just ask Larry a quick question? Could you comment on the interstitial lung disease that accrues as a result of really what is mostly an epithelial process?
That's a great question, and I think those mechanisms are still sort of being worked out. There's some very nice papers from Mike Beer's group in the past year using different mutations in SPC, and there's clearly dysregulated cytokines that are coming from the sick type 2 cells. There's also epithelial loss, and that's probably driving some of the fibrosis from impaired impaired healing and impaired ability to repair. There's also some very interesting work from the German group with ABCA3 on cholesterol, cholesterol metabolism, and ABCA3 may be dealing with cholesterol. And interestingly, one of the things in the differential diagnosis is Niemann-Pick disease. The children can have lung pathology that looks very similar, which is a disorder of cholesterol metabolism. So I think there are secondary hits in the type 2 cell that are leading to changes in the local milieu that are driving the fibrosis and the interstitial proliferation that we need to work on.
Another question here?
Can you speak to how we think about how clinical genetic testing is impacting our ability to learn from these cases? You know, you've been part of that transition and you're still at that interface, but what can we do collectively with this proliferation of labs that are offering panels, results that are out there, and how can we funnel that into the type of discovery that you've led over?
Yeah, I mean, well, you're leading one of the efforts. I mean, one is to collect the information and to sort of aggregate it. I mean, there's a public database, ClinVar, but it's rather sparsely populated. I've sort of tried to make it my mission, at least for these variants, to kind of aggregate and annotate the data, and sort of working with Gary Cutting now in a sort of model to try and model the pathogenicity of them, because the accumulated data really becomes informative. When you start seeing the same variant in trans with other variants, it becomes a lot more persuasive that it's pathogenic than a lot of the other things that are in there in some of the algorithms. So this requires, I think, some infrastructure and some communication, and we can all help one another. I don't know if that answers your question. But.
Francis?
Can you say what are the prevalence of SPA and SPD mutation, and do they have a recognizable phenotype?
Yeah, I didn't touch on SPA because it's, as far as I know, still an adult disorder. There may be one child in one paper. It presents with pulmonary fibrosis and lung cancer, sort of coinciding in adults and familial pedigrees. It seems to be very rare. I only know of about a dozen cases if you go through the literature, but that may be ascertainment bias. It may not have been looked for in patients with the right phenotype. We have looked for patients with SPD mutations and not found any. Interestingly, Susan Wirt, who's one of my longtime collaborators, started staining for SPD in the late 2000s, and we came across two babies who had no SPD staining, and they both turned out to have NKX 2.1 mutations. And then I went back and found a third baby that she'd sent me an email that we hadn't tested originally, went back, tested that baby, and also had an NKX 2.1 mutation. But I don't know of any babies with a primary mutation in SPD in a lung phenotype.
Thank you for that wonderful overview.